Well, good afternoon, everybody. Welcome to the Exeter Waking Up to the Planetary Health Emergency webinar series. I'm Tim Malone. I'm an NHS doctor at the Royal Devon and Exeter Hospital. While people are just coming in from the waiting room, I'll just say that we actually ran the first Waking Up to event as an NHS multidisciplinary educational workshop at our hospital, the RD&E, back in 2006. And that was on the subject of consciousness. And we've been running them intermittently ever since. We run them now in partnership with the University of Exeter since 2014. And they've always been face-to-face events, as this one had been planned as. I can't see you all face-to-face, and I can't talk to you as I normally would in the lobby. So please feel free to use the Zoom chat box to introduce yourself and tell us where you're from. The profound environmental damage we're inflicting on this planet, our common home, is adversely affecting the health of those alive today. And the damage we have already done will continue to impact on human health and well-being for many generations to come. The geo-biological changes we have initiated will persist for many centuries. We're already locked into that change. The actions we collectively take in the next few years will not only determine the future of our own species, but the future of the entire biosphere. That is why we call this a planetary health emergency. The stakes could not be higher. The actions by those of us alive today will determine the future health and well-being for generations of humanity to come, and the survival of many other species on our planet. Our first speaker today is Professor Charles Tyler, who is an internationally leading environmental biologist who played a key role in developing the research field known as endocrine disruption, which is a major issue of concern for both environmental and human health. His research spans a wide range of topics and includes the environmental impacts of chemicals and particles, microbiomes in health and disease, invasive species, and conservation of wildlife populations. Among many roles, Charles is the co-director of the Centre for Sustainable Aquaculture, which is a partnership between the University of Exeter and the Centre for Environment, Fisheries and Aquaculture Sciences. He's also co-director of the Great Western Water Security Alliance, which I always think is something that was sort of set up by Brunel. It's got that sort of name to it. He's received various awards for his work, including the Beverton Medal for groundbreaking research in fish biology from the Fisheries Society of the British Isles, and the Founders Award. I've been practicing that, and I was careful not to say Flounders Award. It's a Founders Award for the highest award given by the Society of Environmental Toxicology and Chemistry. He's published around 300 peer-reviewed research papers. He's absolutely passionate about wildlife, which I suppose is why he was particularly disappointed this year when Freshers Week was cancelled. Charles is going to talk to us about ecotoxicology sitting on a time bomb. Over to you, Charles.
Thanks very much, Tim, and thank you very much for the invitation to speak at this forum. And what I'm going to do today is to, I'm just going to try and change my slides. There we go. I'm going to speak to you about chemicals in the environment as they pertain to environmental health. And for the most part, as Tim said, I'm going to focus on the impacts on wildlife, but I'm also going to touch on terms of impacts in terms of human health, in terms of what we know currently. And I'm going to illustrate to you how we're using and developing some models, zebrafish models, to help us inform how these chemicals cause some of these effects. And so in terms of structure, what I'm going to present today, I'm going to talk, after a brief introduction, I'll talk a little bit in terms of, and illustrate how we're changing our planetary chemistry, and then particularly focus in terms of how chemical pollution is impacting on environmental health. And as I said, in both from a wildlife perspective, but also a little bit in terms of human health perspective too. And then I'll focus very much on the area that Tim mentioned in relation to our work on endocrine disrupting chemicals, a group of chemicals which cause alteration to hormone systems in wildlife and in ourselves as well, and the evidence actually that's impacting particularly on specific wildlife populations. And most of our work in that sector has been in around on species of fish. Then lastly, I'll show in terms of work we're doing in developing transgenic fish. So we're manipulating sort of the genomes of fish to help us better understand how and where chemicals interact in the bodies to induce those health effects. And I'll provide a few sort of concluding thoughts. In relation to the work, and Tim has just really introduced me, really, essentially my real interest really is about integrative research, but trying to better understand how organisms work, how they fit into the environment, and perhaps most importantly from my point of view, how man's activities and anthropogenic pressures impact on those wildlife, both at the level of the individual and of the population. So our work really spans aspects in terms of ecology, understanding sort of bird and other wildlife populations, and the sort of resources they require from the landscape so that we can better protect those features of the landscape in terms, particularly in relation to sort of food resources. It also relates to aquaculture, developing more sustainable ways in which we actually produce food from the aquatic environment. And we particularly focus there on overseas work in Malawi and Bangladesh and India, and especially there looking at sort of how ponds functions in terms of their microbial systems, such that we can enhance those systems, hopefully, and buffering against things like disease, because disease is the biggest factor in terms of sort of limiting aquaculture development. But the mainstay of really what we do and the heart of what we do, I suppose, in the team is really looking at how sort of chemicals and how and the effect mechanisms in wildlife, and to some extent in humans too. And our work really spans everything from microbes right the way through to mammals, particularly focused on fish, and a particular focus actually on the aquatic environment. And the aquatic environment in part because it really is the compartment, the environmental compartment that acts as the sink for just about every compound that we stick out into the environment. And a major part of what we do within the team is developing and applying molecular tools and imaging tools to help us understand how and where these chemicals are mediating their effects. Now, I think we've already seen the last couple of decades in particular in relation to focus on biodiversity, and especially actually in the last 12 months or so, the Attenborough programme and the appeal to the world and to help in terms of protect biodiversity. Biodiversity gives us a fantastic measure really in terms of the quality of the environment in which we live. And as we start to simplify systems, we start to destabilize systems. So for the most part, complexity and richness in biodiversity means stability within our natural systems. Unfortunately, though, if we look in terms of what the data are showing now, for example, here's something called the Living Planet Index, which is a measure of biodiversity, change in average abundance over time. If we look since the 1970s, we see these precipitous declines in aspects of our biodiversity. And it's particularly true for the freshwater environment where we particularly focus. Unbelievably, an 84% decline in our population since the 1970s. Now, this rate of decline in freshwater biodiversity is faster than any other environmental compartment. The sort of predicted extinction rates and to some extent measured extinction rates are operating about 4% per decade in freshwater environments. That's 5 times faster than species losses in any other compartments, terrestrial systems, for example. What I'm particularly interested in fish, basically, and about 1/3 of all the world's fish now, freshwater fish, are classified as either vulnerable or threatened. And there are all sorts of reasons for these declines, of course, in terms of largely imposed, of course, through man's activity, overexploitation, habitat destruction and degradation, but particularly actually pollution. Excuse me. And this translates in terms of biodiversity declines also to the other environments. If we look at terrestrial biodiversity, for example, there's been lots of coverage in the last sort of 5-6 years in particular in relation to impacts of our activities on insect populations, terrestrial insect populations. And there's no doubt across major tracts of the globe, we're seeing these major declines in many of our invertebrates. And of course, the key thing when we look at aspects like this in terms of invertebrates is that they support the higher trophic levels. And so if we look at things like bird populations, some of the best biomonitoring in relation to wildlife population comes from British data. And actually, there's some extensive data for monitoring of things like British bird populations. And again, we can see these major decline in terms of our British bird populations, particular sectors, farmland birds, some of the woodland birds too, and actually particularly some of the insectivorous birds and also long distance migratory birds too. And there's no doubt that some of these effects are being imposed and being caused by exposures to chemicals. So chemicals are playing a major role in terms of decline in some of our wildlife populations. And we're not divorced from these natural systems. And I think increasingly we're starting, I think society is beginning to recognize that we are very much connected to the natural world, the natural environment. We perhaps need to better respect it. I mean, things like COVID-19 have clearly started to heighten that in recent times. But if we look at some of the data and stats in relation to pollution related to sort of human health issues and problems, essentially, It's estimated that about 9 million premature deaths in 2017 were actually caused by some diseases which are related to pollution. That's 16% of all the world's deaths. That's bigger than AIDS, TB, and malaria combined. So some really, really big numbers there. Most of these health effects in relation to pollution come from low and middle income countries. And actually, for the most part, the most vulnerable in our society, in our populations, are the early life stage children, stages in fetal development too. And pollution is hugely costly to us as a society too. I mean, it's estimated in terms of productivity loss due to pollution rest about 2% GDP in these low middle income countries. And health spending varies for different regions around the globe, but up to 7% of GDP is spent on sort of pollution or pollution related diseases in those middle income countries which are undergoing relatively rapid development. Welfare losses are huge, and again, numbers and estimates vary across different countries and continents, but in the US, about 4.6 trillion per year, about 6.2% of economic output. And these health impact burdens are probably underestimated. And it's really just to try to emphasize to you in terms of when we're looking in terms of impact of pollution on the environment, it's not just about wildlife, it's very much about our own health too. And if you're not convinced by those stats and data, you just have to look to satellite imagery to get some feel in terms of how we're changing and manipulating the chemicals or media of our planet. If you look, for example, in terms of the figure on the top left, these are satellite images showing nitrogen dioxide concentrations in the atmosphere. And you can see these high concentrations, the red sort of zones in parts of the US, parts of Europe, and parts of Asia too. And these are at levels in many circumstances at concentrations that we know cause harmful effects to us, our own human health. You can look at other data here, for example, the bottom right in relation to sort of ammonia concentration relating to things like farming practices for agriculture, for example. And again, we can see these hotspots of concentrations around, we have this intensity of human activity. And I think we're all pretty aware and familiar with the issue in terms of ocean acidification, large tracts of our global oceans, which are now having decreasing pH, increasing acidity as a consequence of the oceans are really struggling to be able to absorb all the carbon dioxide that we're putting into the atmosphere. And indeed, our lives are inextricably linked with chemicals. If you look at society as a whole now, we use tens of thousands of chemicals in products. It's about a $3 trillion plus now dollar global enterprise, and they range in all sorts of shapes and forms and morphs, from plasticizers to fertilizers to pesticides to pharmaceuticals. And when we look at these, and I said there are hundreds, there are tens of thousands, and maybe as many as 150, 160,000 chemicals in everyday use. And some of these are what we term high production volume chemicals, so 5,000 or so. And we have concern for these in particular because we are putting high amounts of them out in the environment constantly. And when we look at those high production volume chemicals, only about half have undergone what we might term adequate toxicity and safety testing. And of course, we get exposed to these chemicals, as humans get exposed to these chemicals through all sorts of routes, through our skin, through the food that we eat, through what we drink, and also in terms of things like aerosols. In some cases, chemicals we're taking in through our lungs through aerosols. And just as we're exposed to them, wildlife's exposed to them as well. Now, I don't want people to go away thinking that all these chemicals are harmful. That's clearly not the case. Many of these chemicals we take into our bodies, we metabolize them, and it doesn't do us any harm. But some of these chemicals, particularly those that tend to build up in our bodies, the so-called bioconcentrate or bioaccumulate, they tend to be more harmful to us. And we know that a lot of the chemicals that we're putting out into the environment are problematic in terms of having toxicity to wildlife and or to human health. And some of these are on the so-called priority substance listings in Europe and Japan and in the US and OECD countries. And we try to then restrict their discharge into the environment. And where we shown to be particularly toxic, clearly we try to ban them from use. But there are all sorts of other chemicals which are just coming to the fore now. We're starting to understand that can have not necessarily overt acute toxic effects, but more like sort of chronic longer term health implications. And these are just a few examples, something called PFOAs. These are associated with things like a Teflon, for example, but many other products too. And they cause things like elevations in terms of cholesterol in our own bodies, and actually can also cause things like chronic kidney disease. We have things like flame retardants, which has been well publicized in recent years. And these can affect things like our thyroid function, and thyroid is hugely important part of the body's signaling system in controlling all sorts of physiological processes in our bodies. And then we have things like polycarbonate polymers, things like bisphenols and epoxy resins, and these have been associated with all sorts of health disorders in humans, including actually things like obesity and reproduction too. Excuse me. And then things like pharmaceuticals. Now, pharmaceuticals are somewhat unusual compared to these other compounds and chemicals because clearly they're designed specifically to alter physiological processes in our body. We take them to usually to cure ailments in our cells or our pets or perhaps our livestock. But because they're specifically designed to alter physiological processes, and often those targets are highly conserved, not only into vertebrates, but also sometimes into invertebrates. If organisms in the wild are exposed them at sufficient concentration, the likelihood that their effects are going to be quite high. And then one of these classes of compounds in which this sort of chemicals of emerging concern are these so-called endocrine disrupting chemicals. And this is an area of research we've been involved with for about 3 decades now. And just to illustrate what endocrine disruptors are, if you're not familiar with them, when we think about how our bodies work or how bodies of wildlife work, basically, we have 3 major sort of communication networks. The one is a sort of nervous system, a very fast acting communication system. We have the immune system, of course, which is largely involved with sort of the defense of the body in relation to pathogens like COVID-19 and various other nasty things out there. And then we have the hormone system or the endocrine system. And the endocrine system is a series of glands located around the body. They produce hormones which go into the circulation and into their target site, and they mediate their effects. And the difference between, say, the hormone system compared to the nervous system is that although it tends to be slower acting, if you cause disruptions in that hormone system, they tend to be longer lasting in terms of their effect. And an endocrine disruptor is a chemical or compound, and it can be man-made or natural, that causes interference with that hormone system to affect that signaling process to disrupt things like growth, development, and or reproduction. And it can do that in the individuals that's been exposed or potentially in its progeny as well. And as we're starting to better understand how chemicals work, we're increasingly starting, and the genetics of how the sort of processes work, we're starting to understand that actually some of these effects, through things like epigenetics, can have much longer lasting effects over generations. And so our work in this sort of arena, really, as I started, is something almost at the inception of endocrine disruption. And we were working with colleagues at Brunel University when I was there, and things like CFAS, which had been mentioned in Tim's introduction. And we actually studied wild fish populations, and we found essentially that male fish across great tracts of UK rivers were showing feminization. They were producing actually eggs were developing within their testes, and they were also producing things like yolk protein, which is normally only produced in response to estrogen in female fish and normally laid down in the eggs of the fish. And they had disruptions in something called the development of the reproductive duct too. So these male fish were showing these feminized traits, these feminized characters. And then I had a series of poor PhD students and postdocs who spent many, many years basically doing long-term chronic exposures to real effluent from wastewater treatment works at various sites around the UK to show that actually you could induce all the feminized responses that you saw in these wild fish or these controls to control exposures to these wastewater treatment effluents. So we knew that these feminized responses were coming from exposures to these effluents. Then I worked with some really clever people, some really good analytical chemists. If you're an ecotoxicologist, there's one thing in terms of doing a biology, but you really need to tune up and tie up a really good analytical chemistry to do the sort of like the policeman work, if you like, in trying to identify what chemicals are causing those effects. And we did a whole series of exposures with Liz Hill, and she then took things like the bile. The bile's rather a good medium to focus on in terms of chemicals getting into the bodies of fish, because about 70% of all chemicals are processed through the bile before they undergo excretion. And so what Liz did was we exposed the fish, she took the bile, she fractionated that out through things like HPLC and GCMS, MS, various separation techniques, and she was able to screen those fractions to see if they were estrogenic, if they had chemicals which were estrogen or estrogen-like in nature, using a very clever recombinant yeast screen, which it really went from a yellow to a red if it had an estrogen or estrogen-like compound in it. And then having identified which fractions were estrogens, then she actually went down and actually identified exactly what those compounds, those chemicals were. And actually you can see from from maybe the bottom right-hand figure, there's all sorts of estrogens were getting into these fish. It was like a soup of estrogens. And it included things like estrone and estradiol, steroid estrogens that emanate from women. It included things like the components of the contraceptive pill, ethinyl estradiol. Also included things, strangely, things like horse estrogens, believe it or not. And some horse estrogens are actually used in things like hormone replacement therapy. It includes some industrial products, surfactants, non-alphenol and its polyethoxylate. It includes some phthalates and various other plasticizers too. Lots of chemicals were getting sucked up into these fish, and likely these feminized responses that we're seeing was a consequence of this mixture of these estrogens which the fish were being exposed to. Having said that, some of the compounds, and notably one pharmaceutical, we found to be exquisitely potent in terms of inducing these feminized responses. And going through some PhDs and postdocs with long-term exposures of roach under controlled conditions to ethinyl estradiol, we were able to show that we can induce all of those feminized responses, all those feminized phenotypes from exposures to ethinyl estradiol at concentrations that we find in the environment. So you could reduce the yolk protein production, you could reduce these oocytes within the testes, and in fact, at only 4 nanograms per litre, which you would probably find in some of the most polluted effluents, you can cause complete sex reversals. So that exposure throughout the life of the fish caused males to turn into females, essentially. Excuse me. And one of the big questions in all this, so okay, so we're seeing feminized responses in these fish. Does it really matter really to the individuals in terms of, does it affect their abilities to reproduce? Clearly, if you switch the male to a female, it does. Well, what about if it has some oocytes contained within the testis? And then we did some controlled studies whereby we actually took males and intersex males, feminized males from the wild and females, and we put them in these breeding groups of competition between males and females, with 3 females, 6 males of various degrees of normality, if you like, in terms of feminization. And we developed some genetic probes through something called DNA microsatellites, so we could genotype the parents and then also genotype the offspring that they produced. And from that, we could match the offspring, the babies, back to the parents, the mums and dads. And then given the fact that we knew whether males were feminized and to what extent they were feminized, we could map reproductive success against this so-called feminized level of feminization, what we call the intersex index, essentially how many oocytes there are in the testes. And you can clearly see from this that as you get an increased degree of feminization of those males, they were less able to sire offspring under those competitive breeding scenarios. So from this, you can clearly see that males which are feminized or moderately to severely feminized for sure have a reduced breeding capability. And since then, we've gone on and we've done all sorts of population level studies looking at things like whether there's been changes in genetic structure of those populations. We're also looking to see whether there are changes in terms of the genes involved with those estrogen pathways. And we're not alone in terms of our finding at all in relation to these findings in fish. And so all sorts of other studies now have been done, all sorts of other wildlife, from alligators. Good friend Lou Gillette in the University of Florida did all his work on alligators, in fish-eating birds like grebes and ospreys, for example. Otters have been shown to have reduced penis size in parts of the US related to chemical exposures, immunosuppression in some of the pinnipeds and some of the seals, and some really interesting work coming out from things like studies on amphibians and on birds, where they're showing that some of these chemicals are altering things like song and song pattern. And if you're a dull bird like this wonderful bird here, or if you're an amphibian, song is everything in terms of the male, in terms of sort of commanding a territory. So some really interesting, wider, more pervasive effects of some of these endocrine disrupting chemicals on wildlife is coming to the fore. And clearly, as humans, we're not divorced from these effects either. And actually, even though some of the original data in relation for endocrine disruption really came from studies on wildlife, it wasn't really until the studies on humans in relation to things like falling sperm count came to the fore that actually this really got sort of widespread news, and certainly in terms of sort of press coverage. And now from the very detailed and extensive studies largely on sort of rats in the laboratory, but also some epidemiological studies, there's very good evidence that some of these chemicals are causing adverse health effects in humans too, including things like obesity. Some of these chemicals are what we term obesogens. They serve to drive things like fat deposition, and some really interesting work on humans too, which parallels that in some of the wildlife. There's some of these behavioral and learning disorders associated with some of these chemicals too. So one of my real interests from all of that fish work, which was largely focused on one group of endocrine disrupting chemicals, these so-called environmental estrogens, is that estrogens in the body, I think most people associate them as a female hormone, essentially, in terms of controlling sort of reproductive cycling. But actually, estrogens do all sorts of other things in our bodies, in both males and females. They're involved with growth, bone development, calcium homeostasis, defense, immune function. Estrogen signaling is really complex. There are a number of different estrogen receptors, 2 in humans, 3 actually in fish. And there are nuclear receptors and membrane-bound receptors, the crosstalk between those receptors as well. It's probably the case that every cell in the body expresses an estrogen receptor sometime during its ontogeny, sometime during its life. So our sort of interest, subsequent to those findings on reproduction, well, if these estrogens are having that effect on reproduction, and we know that in mammals and in human estrogens play all sorts of other roles, how are they actually affecting other parts of the physiology of our fish? And so I teamed up with Tetsu Kudo at University of Exeter, and we thought, well, how about we try to develop some transgenic fish so we can better visualize how and where these estrogenic chemicals are interacting in the body. And I won't go into all the technical details today, but happy to talk to any nerds in this sector afterwards. I'm one, basically. But here I'm illustrating the first transgenic we developed for assessing exposure to environmental estrogens. And it's a sort of double transgenic that enables you to get an amplification in that signaling when an estrogenic compound interacts with an estrogen receptor. So now what we had this biosensor whereby we could track responses in the fish in almost real time. So every time a chemical interacts at that receptor site, that receptor interaction site, you get something called a fluorophore produced. In this case, it was a green fluorescent protein, which you can then visualize through imaging. So you can measure responses in different body tissues. You can measure responses to different concentrations. So you can look at which tissues respond and what sort of the relative sensitivities of them are. So using these biosensors, now we have this great utility, we have this great tool to be able to start to better understand how and where these different types of environmental estrogens are interacting in our exposed transgenic zebrafish. And these are just some profiles now with different concentrations of different environmental estrogens. So I've talked about ethanol estradiol, that exquisitely potent estrogen. Here's a surfactant, non-alphenol, that we find in lots of detergents and paints. And we can do it also for natural estrogens, things like genistein. And one really interesting one, which has a real sort of good and interesting link, if you like, in terms of human health, is that for bisphenol A. And what we found with bisphenol A when we were exposing is that we were seeing responses in particular in relation to the heart. And illustrating the power of these transgenics in terms of being able to refine and hone down into how and where these chemicals are interacting and what it might mean in terms of health. We went on to look at the heart, basically, in terms of its response to this particular environmental estrogen, bisphenol A. And what we're looking at here is actually the heart that's now been dissected out from our exposed zebrafish, and the red shows the myocardium that's been stained with a particular antibody, and the green showing where in the heart that it's responding to estrogen. So the top panel's showing for bisphenol A, and the bottom panel is showing for one of its metabolites, its main metabolite, its main breakdown product, something called MBP. And what you can see using this transgenic is that actually the bisphenol A isn't interacting with the myocardium per se, it's actually interacting with the valves, the outflow cushion and the AV valve. So that just shows you the utility of these models for us to be able to hone down and find out exactly where those interaction sites are. So potentially what the health implications might be. And in fact, we've gone on, and Ross Brown and the team has gone on to look at the functional link between exposure to this particular compound and the ability of that heart to function in life. Now, I'm very conscious that in terms of, when we think about exposure to chemicals in the environment, that endocrine disrupting chemicals much more broadly, we're exposed to complex mixtures. And that's true for humans as it is for wildlife. And I'm sure you could talk to many people in the Med Center at Exeter and further afield, and they'll tell you in terms of, in particular in relation to early life stages, which tend to be the most vulnerable to some of these chemicals and their effect. If you take measures from things like cord blood, for example, you can find in lots of those chemicals that I've already mentioned, some chemicals we know that are problematic to our own health. And it's exactly the same for fish. If you go and measure things like concentrations and the variety of chemicals in the livers or in the bile, these tissue processing stations in the fish for chemicals, it's almost like a blueprint for what we're putting out through effluent discharges. You will find antidepressants, you will find anti-inflammatories, you will find endocrine disrupting chemicals, and all sorts of other xenobiotics too. So these fish are being exposed to complex mixtures of chemicals. And what we want to know really, of course, is in terms of what the effects of those integrations are. And so what we're trying to do with these transgenic models now is, rather than have a single specific site of interaction for chemicals, we're trying to develop these models whereby we've got a number of interaction sites for different types of chemicals or different types of response mechanisms. And the idea is to try and build up these biosensors so that they're more complex and we can better understand how they work in a much more integrated manner. So we have them not just for regions now, but for oxidative stress, for immune function as well. And there's one other thing in relation to mixtures I just wanted to say, is that although we think about being exposed to mixtures, another key thing that we need to consider is that in terms of the history of our exposure. Because so we've developed this little transgenic now whereby this is for the estrogen transgenic, and rather than having that green fluorescent protein, we've actually got something called qaeda. We just replaced that fluorophore essentially, and qaeda also turns green if that interaction site is responding to chemical exposure. But if you expose it into UV light very briefly, it'll go red. So now we have a system that we can do sequential exposures to see how one exposure affects the responsiveness subsequently to later life exposures to those classes of compounds. And what we're starting to find is that really matters in terms of response sensitivity. So again, it's just to illustrate to you the models to help us better understand what sort of lifetime exposure and sequential exposure to some of these compounds might mean. It's also absolutely key in terms of these models. We're trying to find that there's this crosstalk between the endocrine system and the nervous system. So we've illustrated that some of the neural tissue, both central and peripheral, responds to these estrogens. So it has these estrogen response activity. And a very talented postdoc in the team, Aya Takazana, has recently identified that there are some smell cells and something called estrogen responsive olfactory bulb cells in the brain during early development in the fish, which fundamentally require estrogen and estrogen signaling appropriately to actually develop to facilitate smell. If you disrupt that or cause exposure to environmental estrogens, you can disrupt the ability for them to smell in later life. And that's bringing me to a close now. And that's really what I wanted to try and emphasize in terms of some of the work that we're doing, is that we often think in terms of how chemicals mediate their effects in our body in silos. It's either it's an immunosuppressant, or it affects a neural system, or it affects the endocrine system. In fact, there's so much crosstalk between these signaling systems that if a chemical's causing a perturbation and a disturbance to one part of that signaling system, it's quite likely it's causing perturbations across the system. This is my last one in terms of these particular zebrafish models, just to, again, to try and illustrate the utility of them in relation to thinking about how chemicals mediate effects in the body. This isn't one that we developed, it's one developed in the US, but one in particular we're using it. Matt Winter in the team who's been pioneering some of this. And this is a zebrafish, which it's a transgenic whereby there's been a link in terms of a fluorophore to the neurons in the brain. So if we just see these two little videos here, this first one is just showing a zebrafish. This is a live zebrafish at rest, basically, and you can see a little bit of activity firing away in the brain. If we now expose it to a chemical, this is actually a seizurogenic compound, we can start to see all this activity across the brain. So we can start to chart and map where these chemicals are interacting in the brain. And Matt's doing some wonderful stuff now in terms of trying to link in terms of those signaling processes across the brain, and how that through something called connectograms, and then link that to sort of behavioral phenotypes. So we have, again, this great utility to better understand how chemicals might affect things like behavior. So concluding now, I hope I've illustrated to you that pollution is certainly a major environmental threat to humans and wildlife health. Chronic exposures are something we need to get a much better handle on, certainly in relation to effects over generations, because they're really poorly established. When we think about effects analysis, we definitely need to be, better consider environmentally relevant exposures, things like timing, mixtures, interactions, and other stresses. And I hope I've convinced you that these little transgenic zebrafish models have fantastic utility for combined with imaging to help us better understand how chemicals interact in the bodies and to better inform more intelligent approaches to think about sort of health effects might be for human health too. And lastly, I would say, and I think this echoes sort of, at Tim's meeting today, is, we humans really aren't that much different from fish. We're not divorced from the environment. And there's some, wildlife can act as fantastic sentinels to help us understand the potential impacts of chemicals on our own health. And lots of people have funded this and contributed this, thanks to them, of course. I hope you're still awake. I can't see you, basically. And I think I'll take questions probably after David's talk. Thank you.
Thank you very much, Charles. That's an amazing run through multiple subjects there in ecotoxicology and analytical chemistry and biology. We'll move straight over to David's talk and take questions for both talks at the end. Just while we're changing slides, I just wanted to tell you actually something. There's a lot to reflect on in Charles's talk, and I wanted to mention the word riptide to you. So that's the word that's of the day, riptide. Keep that in your memory. Climate Matters is a publication, an online publication, and will be in print later on in December. It's a collection of poems, stories, science writing, and artwork, which is launched online today as part of the Waking Up to the Planetary Health Emergency webinar series. And it questions the climate crisis, COVID-19, capitalism, health and destruction of our planet. Resounding voices are calling out, we need to act now and to act fast because our survival depends on it. And we're going to have later on in this series, and I hope you'll be coming back for the other episodes of this series, there are 6 in total. We're going to have some recorded recitations and readings from some of the poems by some of the authors in that publication. And we'll give you there'll be a link to that on the chat. And I've had a brief sort of preview look at it last night, actually, and it's an amazing publication. It very much makes this into a transdisciplinary workshop, which is actually what we're trying to achieve. So we're going to move on to David Butler now. David is Professor of Water Engineering and Head of Engineering at the University of Exeter. He's an internationally leading researcher, teacher, manager and consultant in water care, in the water sector. Has a degree in civil and structural engineering from Cardiff, and both a master's in public health engineering and a doctorate in environmental engineering from Imperial College London. He's a chartered civil engineer, a fellow of the Royal Academy of Engineering, and the Institution of Civil Engineers and the Chartered Institution of Water and Environmental Management and International Water Association. David is going to tell us about water, friend or foe, how to build local resilience to global threats. Thank you, David.
Thank you very much indeed, Tim. Yes, I'm going to give you an engineering perspective. So I feel a little bit nervous on that, presenting to health professionals, but bear with me. And I really want to talk about water and the engineering of water under global threat. So the talk will cover briefly global threats, then try and answer that question, is water a friend or is it a foe? What is resilience? The safe and sure framework that we've developed, and then looking at options for building local resilience. Now, I pulled this piece of information, this study recently, which was carried out in 2019, and it was an attempt to rank for a number of global cities the economic effect of global threats. Number 1 out there at the top there comes out at market crash. You'll notice that human pandemic is way up the list, and we know where we are right now. But the reason I pulled this diagram up was just to flag up, the very next one is a watery one, flood, drought and freeze. So, thinking about global threats, water is way up the list in terms of its potential and its potential threat. So to that question then, water, friend or foe. I mean, we all love water, don't we? You know, we're drawn individually to water. We go to the seaside, we bathe in water, etc., etc. And we love to see pictures like that. This is a reservoir in the north of England, a beautiful setting. But of course, reservoirs don't always look like that, you know, and increasingly common, we see a reservoir that looks like that. And, you know, it's not so long ago, is it, that we saw this particular situation in Whaley Bridge, where the spillway to the dam had failed for various reasons, mainly poor maintenance over the last number of years. And we have a lot of aging infrastructure. Then again, how about this picture? Another lovely shot. This is the River Exe, actually, in the Exe estuary, Topsham there, and Exmouth out in the distance. A beautiful setting. Again, we're drawn to the seaside, but it doesn't always look like that. This is a shot of the River Exe in Exeter in 1960, and very extensive flooding. Flood defence works have been built since then, but this is a shot of Exeter just a few years ago, showing the River Exe just on the point of overflowing and overtopping through Exeter. So water is a friend, but it's also a foe, and we have to live with those tensions, and that's what we really do in the water sector. But from a global perspective, and as a water engineer trying to deal with these effects, for me, the key issue is variability, and that's what we're seeing, unprecedented variability. We find it very difficult to use our historical models of rainfall, for example, which we've traditionally extrapolated into the future on a nice straight line. We can't do that anymore, because what's happened in the past is not necessarily going to happen in the future. And as we know, you know, 1 minute it can be flooding, and the next minute we can be in a drought. How do we deal with that? Well, we deal with it by grappling with the notion of the idea of resilience. I guess we've probably all got our ideas about what resilience is in our own sectors and in our own lives, to be honest. But I wanted to just pull up three not world leaders, but one world leader, and some very important people to give you their view, I think, of resilience. And Nelson Mandela, he said, "Do not judge me by my successes. Judge me by how many times I fell down and got back up again." And to me, that's an important message about resilience. It's about falling down, but when we fall down, getting back up again. And can we do that continuously? Next, Nicola Adams. Now, here's Nicola Adams in the Commonwealth Games, I think, some years ago. As we know, Nicola is now strictly-fied, and we see her doing that. But what she said when she was a boxer was, it's not how you go down, but how you get up. Another really important facet about resilience. How does a system that has been shocked, that has been dented, has been punched in this case, how does it get back up again into action? And then the final key individual is Tigger. I don't even need to introduce who Tigger is to you, but what Tigger had to say was, life is not about how fast you run or how high you climb, but how well you bounce. And in my opinion, resilience is about that bouncing. It's about that bouncing back, or bounce back ability, we like to call it. And it can even be about bouncing back better. So I was fortunate enough a few years back now to have a fellowship, a 5-year fellowship, a so-called established career fellowship. And what that was to do was to develop a new idea, a new paradigm, a new method for managing urban water in the UK and elsewhere, really to deal with these challenges and global threats and uncertainties. And I called that safe and sure. And what did I mean about that? Well, by safe, I meant reliability. That will be another word for safety. And by sure, I meant sustainable and resilient. So traditionally, from an engineering background and in our engineered systems, we have developed systems where safety is paramount and safety is baked into the system. And there's nothing wrong with that whatsoever, except that we might be building safe systems that worked 20 years ago. Are they going to work into the future under all these uncertainties? And therefore, I was looking at how we could go forward to also build resilience and also build sustainability, understanding that sustainable systems built on resilient systems that in turn built on safe systems. One of the things that came out of that fellowship was what we called the Safe and Sure framework. This is a theoretical framework, a conceptual framework, but has proved to be very useful in understanding and relating the relationships between these global threats and uncertainties that we've been talking about with the system that we're interested in. Then when that system is in trouble and stressed, what the impact of that is, and then subsequently what the consequences are. So for example, one could imagine that urbanization is a global threat, a creeping global threat. And in my world, in the water world, what's the implications of that? Well, even for the same rainfall, even if that wasn't changing, the more you seal the surface, the more you generate more flows and so on. And that's going to increase the flows into our system. In our case, a drainage system or sewer system. But that's an existing one. That's been built with safety in mind, has a fixed capacity. And if we stress it, it's going to fail. And when it fails, it overflows like this. And ultimately, the consequences are this. These are not Charles's fish, but these ones essentially have been suffocated because of the organic load caused by these overflows. So by using this type of approach, we're able to think quite carefully about these interrelationships and what we can do about them. And we've identified the intersections between the threat and the system. We can mitigate that threat. So the idea then is if we mitigate the threat, we reduce the threat. So in our case, we might mitigate the threat by softening the surfaces, by making them greener, for example, and therefore reducing the amount of water running off. This intersection here is about adaptation. How can we adapt our system such that it fails less often or less severely? So we're trying to reduce the impact of failure. In this case, it might be building bigger storage tanks and so on in the system. And what about when that system actually does fail? And if it does, this intersection is coping. How can we develop coping strategies to reduce the consequences? And in this case, for example, we might want to try and push oxygen into the river on an emergency basis in terms of improving the water quality and maintaining the water quality in the river. And the final one, the final intersection there between consequences and threat is learning. We need to learn from our experiences. What have we been doing with this framework? Well, we've worked with Scottish Water, and we've developed a model, a simple, relatively simple spreadsheet model that allows colleagues there to look at the various ways of intervening to improve resilience and help them in their engineering schemes. We're currently developing a mobile phone app, actually, with Northumbrian Water. And there they're using the framework to look at organizational resilience. How can resilience be developed in the organization as a whole? And finally, COVID-19, we've been doing a survey with the water industry as a whole and looking at ways in which it's mitigated, adapted, and coped to and learned from all the things surrounding COVID-19 in the water industry. But I was keen, just in a few minutes, to move from the kind of conceptual and theoretical down to something a bit more practical, and to tell you about some of the work that we've been doing right at the local level, building local resilience. And one of those is rainwater harvesting. Most of you will understand what rainwater harvesting is. We collect water from a roof, drain it down into a typically underground tank, then that's pumped back up to some sort of header tank in your loft. And there you can use that water for non-potable applications. Advantages, well, you don't need, therefore, to use so much drinking water for flushing loos and that sort of thing. So a definite advantage to reduce drinking water. If the system fails for any reason, maybe there's a burst outside or in a drought situation, you have an emergency supply, so a very good resilience tool. And with careful design, we can also use them to reduce flooding downstream if we can manage to use the storage appropriately. So they do have some very significant advantages, rainwater harvesting systems, but, and the but is, they are quite expensive and you've got to run them. So you need a big hole to put your tank in, which is quite expensive on an individual house basis. And also they require energy because you're going to be pumping back up here and therefore generating greenhouse gas emissions and indeed contributing to the thing you're trying to adapt to or mitigate. So we've been working on different systems to try and do that rather better and rather more efficiently. And here's one idea. I don't know if you can see it in the picture here, but it doesn't involve a great big tank underground. We've moved the tank and we've put much smaller tanks actually on the downpipe, on each downpipe of the house, much higher up. And in the smaller tank there, the bulbous downpipe, there's a reduced amount of storage, but we take our pump lead from here and put that into the loft. And here's a picture of one of the tests that we've been doing. We took 3 houses just in Exeter and built some of these systems. So the small pump sits in your loft here, and when the small storage outside fills up during rainfall, it's pumped into a header tank there and then used internally. And what we found was that sort of system, we can gain benefits in terms of reduced water use, so a reduction in costs. But there is some additional cost because you're going to run your pump, but really quite low because it's a very small head difference now. You're not pumping from really underground, but from a much higher level. And here's another version, another idea that we were developing, which uses no pumping whatsoever. I don't know if you can follow what's going on. It's a rig built in the laboratory where we're putting rain on a mock-up roof, and we rigged up a system of tiles that have a slot in the top of them. So water draining off the roof now goes into those slots and then is channeled away, and there it is going into a high-tech bucket there you see on the floor. But in principle, what that's showing is you can trade off a bit of roof, but you can therefore using this system, you get rid of pumping altogether. And my final slide, what's the future of this? Where are we going with these systems and so on? And well, that's smart. The future is here now. We've been developing systems that link our storage systems and our collection systems now to the cloud, not the physical cloud, but the digital cloud. And you can see on the left here, we have a system that's been built outside this particular building, but it's got a tank that's not underground, it's above ground, and we've linked that up to our system. And we can poll that and look at the performance of that from anywhere because it's linked to Wi-Fi. Here's another version on the right here, which is similar, but that's got solar panels, so that reduces our pumping costs as well. And the future of these sorts of systems is manyfold, but includes linking up to weather forecasts, so we can empty the tanks prior to a storm and hold water back and reduce flooding. It also potentially allows all of these individual systems to be linked up such that we can take an overview and maybe even play tunes. If you're a water company, you could open some tanks and close others into the future and so on. So my summary and conclusions then, I would say water is both our friend and our foe, but it's the unprecedented variability that's really stressing us as engineers and challenging us what we need to do into the future. This idea of safe and sure provides quite a useful framework, we think, for linking threats to consequences, and more particularly for identifying interventions where we can increase resilience and sustainability. And that framework is generic, actually. It doesn't have to be anything to do with water, and is already proving that it's quite a useful framework for other applications. And then finally, rainwater harvesting provides one way of building local resilience to global water threats. We think that's got great potential. So thank you very much indeed.
Thank you, Charles. Thank you, David. That's fantastic. We've got a few minutes for questions. So if Charles is also there and will put his microphone on, please put your questions in the chat function. I'm going to start with Charles. We've got a question for Charles. And I think one of the issues you illustrated there was the comprehensive, the sort of chemical soup that we're all being exposed to, or wildlife is being exposed to at the moment. And it reminds me of what's going on with COVID-19 at the moment. It's all about the receptors on cells where the actual chemicals are actually targeting. And we probably at this stage I don't know what it's like in the fish world or the biological world, but we probably don't know enough about where those receptors are and what cells they're actually on. There's a project in medicine called the Human Cell Atlas, which is actually trying to identify the different cell types for all the different organs of the body. And surprisingly, we don't know that. Are there parallels in fish research and aquaculture?
Oh, for sure. I mean, there's issues in terms of, clearly some chemicals don't interact with one receptor, they interact with many different types of receptors. And so, so they're unlike such, some of the hormone mediated process, serrations, for example, there are some receptors that will receive a whole variety of different compounds and chemicals. So it makes it very complex in terms of understanding the specificities in terms of sort of the chemical signaling processes for some of these environmental contaminants. It's also it's also the case that not all processes are exactly the same or parallel across different organisms, of course, and so some organisms tend to be more sensitive than others, and of course you have differences in terms of things like niche. One of the biggest challenges here really in relation to human and wildlife health is, touching on your point really, is in terms of what are those target organs, and really trying to better understand the fate of these chemicals in those different tissues is a big challenge. And I think some of that research will better help identify where we should be more focused in relation to think about health implications.
Thank you. And there's a question in the chat here that somebody specifically asks about glyphosate as an endocrine disruptor. Any tests done specifically on that?
That's a very interesting one, and it's a very pertinent point, I think. And glyphosate is a very controversial compound, I suppose, really, in terms of its possible health effects, both in wildlife and potentially in humans too. And I think the jury's still out, I think, is in terms of being an endocrine disruptor. Many papers now are showing it can be a weak estrogen, a weak anti-androgen. It can interact with the thyroid receptor, etc. But in terms of in vivo studies, again, it's sort of a mixed picture. And I think it's in part because, or two reasons. One is I think glyphosate can interact in many parts, many receptors in the body, and so you can get all sorts of different effects. And where they're specifically endocrine or more sort of like low-level sort of overt toxic effects is questionable. Also, a lot of those studies, original studies at least, were done on glyphosate formulations, so things like Roundup, etc. And they have all sorts of other chemicals contained within them as well. So I think the picture's still a bit blurred there. I mean, I don't think it's a good chemical to have out in the environment, but in terms of its relative toxicity compared to others, it's still a little bit questionable.
Thank you. David, a question. I mean, I love the way engineering is always, coming up with solutions, very practical talk and practical solutions with all the challenges that we're facing. Are you going to be able to keep up with the variability? I mean, the variability seems to be the trouble here. That things are changing so fast in the natural world that we can't necessarily find engineering solutions in the time that we have.
Yeah. You're absolutely right. I think engineering can go so far, but I think we have to look quite holistically, and that's why we kind of, the thing we've come out with the safe and sure. You need a raft of measures. You need to mitigate threat. You need to adapt your system. You need to have coping strategies ready, and you need to be able to build on what you've done and learn. And we were very keen when we're using that to apply it with the water companies to look at that, to see what they got, what was their portfolio, because traditionally it had been very engineering based, has to be, but what else is there, and what are their strategies going forward?
There's a couple of questions in the chat, which I think for both of you really, is about water filters. I mean, one thing when I was looking at your picture, David, of the recycling the roof water, you had a filter on there. I was just wondering how that actually works and what what kind of things are filtered out from the?
Yes, yeah. Well, it's non-potable. We're using for in these typically non-potable applications, so they're just coarse filters. We have done some work previously with a company that had produced a rainwater to drinking water system. And actually, any of you that know the university campus, this machine is still in the innovation building, in one of the plant rooms, and you may not have noticed that we had it working at some point, and some of the water filters were actually connected to this thing. So you may have, if you've been on campus, you may have been drinking rainwater. But the reality of that is it becomes very expensive. So we can turn rainwater to drinking water, but to do that successfully, it becomes much more expensive than doing it, using a traditional system. So you've got to hone your system to the application, really. And we think the most cost-effective way, domestically anyway, is to use it for non-potable application.
And what about the problems of storage of the water in your loft during the hot summers and the...
Yes, yeah, that can also be an issue, absolutely. And we have done some work on that as well. Making sure the water quality doesn't degrade. Same thing in the innovation building on the university campus. We looked at that in some detail, and associated with that is do we get biodegradation of going on within the tanks and so on. The one thing we found was a bit tricky was if you left the system for a long time and did not use it. So, went on holiday or something like that. So the solution to that is to have a pre-programmed emptying function that automatically emptied the system after a week if it wasn't used, and so on. So there are kind of practical solutions.
Thank you. I mean, Caroline Jessel, I noticed in the chat, has asked, I think perhaps to you, Charles, about all the chemicals in the water. Is there any way that these chemicals can be removed? Can they be filtered out? Are there any chemicals that, the examples you gave all have adverse effects on fish and on human health. Are there any good chemicals that we put in the water that actually have some beneficial effects for us?
I'm sure there are, Tim, but it's not ones I particularly focus on. I'm sure there are, and I'm sure some of the sort of possibly low-level exposure to some of these agents might be boosting some cell function and capabilities. In fact, there was someone in the world of sort of toxicology many years ago who said that we're exposed to all these harmful chemicals, we're equally exposed to many good chemicals, and the two will balance itself out. I don't run with that sort of idea or hypothesis, basically, but I'm sure there are. But in terms of thinking about filtering them out, yes, of course, in terms of some of these water filters, very effective at removing some of these harmful compounds and chemicals. Equally, I would say in terms of some of the filters that are actually put in, they have things like phthalates and things in them which actually leach out into the water. So, yeah.
Thank you. Now we've run over a little bit. I'm happy to go on for a few minutes if people have got more questions, but I understand that some people will need to leave the webinar now. Please continue to write your questions in the chat box. For those who need to leave, you will get an email with a Q&A, a bit of feedback on the session. I'd be very grateful if you could actually fill that in on the Google form, and so that we get some feedback from the many of you that have been here. And we hope to see many of you again next week, same time at 12:30, for the second webinar in the series. And the series will run for over 6 weeks. I wanted to ask Charles a little bit more about the combination. It was interesting what you were saying about the combination of different chemicals and a lot of drugs that you were on your slide there. And one thing I feel very, very conscious about as a doctor is that we learn all about the pharmacology of these drugs, whether absorbed into the body and how they might affect us, but we learn nothing at medical school or about what happens to them when they're excreted. And excreted they are in, as you were pointing out, huge quantities. I mean, that's a failing in our education, a failing in our sustainability, a failing in our approach to how we use pharmacology, is it not?
It is absolutely so. I think the sort of life cycle analysis in terms of embedding that further in the education programs much more broadly, and especially, as you say, into the medical sector, in terms of, I think, for the most part of the time, when you talk about pharmaceuticals, they're largely directed to improving and aiding human health. And we don't tend to think about environmental consequences of their use at all, to be honest with you. And I actually come from a bit of a medical family, actually, and even my parents were quite ignorant to this. And it is very much about trying to get better education, from the outset into those sort of systems. So it is better embedded. Of course, things like antibiotics use, of course, have served to heighten our consciousness in this sector, of course, in terms of AMR. But that's in part because that's clearly coming back. We know that's going to have major implications for our own health, basically. So unless things are really sort of, once you start to move away from direct immediate impacts in terms of human health, there's less understanding, there's less education in that sector, and we really need to incorporate it more fully.
And it's often in certain corporate interests to not inform us or not talk about that sector.
Oh, of course, Tim, and I think the pharmaceuticals is a big industry. I'm not sure it is in the UK, maybe 2 or 3% of UK GDP, and so they're incredible, powerful organisation lobbies, basically.
David, can any engineering solutions in terms of smart sewers and monitoring of sewage and seeing what's actually in there and therefore, being able to divert some of that into processing or diverting it away from where it may get into be exposed to wildlife, and also while you're on that subject, we can't in the middle of the pandemic move away from sewers and not mention monitoring of COVID-19. Do you want to just cover those two topics?
Yes. Well, yeah. It's a great thought, isn't it? Smart sewers, and they are getting smarter. Actually, I think what we're doing in terms of smartness is understanding exactly what's going on and how they're functioning and so on a lot better than we used to. It was essentially getting it in a pipe and getting it away, but we've developed a much better handle on how they operate now. We're able to measure the flows, how full the pipes are, the big ones anyway, and take appropriate actions. And that can be as simple as putting them into storage, or it can be, because typically, as you saw from some of my pictures, a sewer system can just overtop when it gets too full, and we want to try and avoid that as very much as far as we possibly can. But the real solution, frankly, is to stop the flows getting into the system in the first place, because once they're in there, we have to handle them and do something with them. And that's why most of us are very keen to look at how we can do these upstream things, like hold water back right up in the city. So in rainwater harvesting, for example, I didn't talk too much about it, but if we can make those smart and hold the water there, then it doesn't get into it. We can drip feed it later on. And yes, COVID monitoring in sewers, number of colleagues around the world are doing that now. Not an expert on it myself, but talking about excreting, and certainly that has got an interesting angle to it in terms of trying to localize where outbreaks are and so on. And I know a number of the water companies are undertaking trials on that right down at their sewage works and monitoring levels, virus levels.
I think one of the early pilots was in the southwest, was it not?
Exactly right. Yeah, that's right.
A final question, probably for both of you. Actually, somebody's put it in the chat, and actually, it's a good question to ask. I'll go to Charles first. Charles, do you still eat fish? Are you happy to eat fish?
It's a question I often get asked mainly by the media, actually.
Okay.
Yeah, I think when you look in terms of concentrations of these contaminants, actually, for the most part, they're not in the musculature. They tend to be in tissues like, for example, the liver or the gut or the brain. So actually, some compounds clearly permeate into the parts of the organism that you eat for the most part, but in terms of muscle, they tend to be at pretty low levels. And also in terms of when you look at the concentrations across many environments, they really wouldn't be in that musculature at concentration to give you human health concern.
And David, is fish on your menu?
Well, if Charles says it's okay, then it's fine. Yeah, it's on my menu.
You wash it first, though, of course.
Of course.
Some portable water. Thank you very much. Thank you very much indeed, both to Charles Tyler and David Butler. That was a fascinating run through and so much to think about. Please join us next week, 12:30 on Thursday, the 12th of November, when we'll be looking at, I think, the economy and what we can learn from history. Thank you and goodbye from Exeter.